So welcome once again, and today we are going to start with the new disease, that is rheumatoid arthritis. Now let us see the definition of this disease. Rheumatoid arthritis is a chronic systemic autoimmune disease. So it is a chronic disease, it is a systemic disease, means it is involving multiple systems there, and it is an autoimmune disease. That means in this disease autoantibodies are formed, and these autoantibodies they are going to act, or they are going to attack our own tissues. We will see that what what is the what are the tissues which are involved with this. So it is a chronic disease, it is a systemic disease, it is involving multiple system, and it is a autoimmune disease characterized by persistent inflammation of the joint. So what is the characteristic finding in this patient? Persistent inflammation of the joint. Joints are persistently inflamed. Now in this disease, autoantibodies which are formed, what they do? They attacks the synovial membrane. They attacks the synovial membrane, leading initially to pain, swelling and restriction of the joint movements. So these autoantibodies which are formed in this disease, they are attacking the synovial membranes of the joint, and initially what are the clinical features we are getting? We are getting painful joint, we are getting swelling of the joint, and we are getting restriction of the joint movements. And latter here means, once the disease enters into the chronic stage, it leads to bone erosion. It starts bone erosion, and finally the patient develops a joint deformity. So because of this bony erosions, finally the patient gets joint deformities. So let us try to read it again. It is a chronic disease, it is involving multiple systems, and it is an autoimmune disease. In this disease autoantibodies are formed, and what is the characteristic finding in this disease? Persistent inflammation of the joints, and the autoantibodies, what they do? They attack the synovial membrane, and initially in this disease we are getting painful joint, swelling of the joint and the restriction of the joint movements, and once a chronicity is achieved, it leads to bone erosion and patient develops joint deformities. Let us see some interesting facts regarding this disease. This rheumatoid arthritis is more common in females as compared to males. Almost of the total patients, 70% of the patients they are females. Then a common age group of onset of the rheumatoid arthritis. So common age group for the onset of the rheumatoid arthritis, it is in between 40 to 60 years of age. So these are the some interesting facts with this disease. Now we will see the etiological factors of the rheumatoid arthritis. So here I have mentioned the three factors. So let us first see these factors. So first factor is an environmental factor which includes smoking and infections. Of these two environmental factors, this smoking is very important, and this smoking is been considered a very crucial factor of responsible for the rheumatoid arthritis or in the pathogenesis of the rheumatoid arthritis. And the second factor is the infections, and in bracket what I have mentioned is the mucosal infections. So here mucosal infections means the infections which are related with the mucosa of the respiratory system or a periodontal mucosa, or even the mucosa of the GI tract. So, there may be the infection of the GI tract, there may be the infection, periodontal infection, or there may be a respiratory tract infection. So, mucosal involvement of these various mucosa, they may be involved as far as these environmental factors and the infections are concerned in a rheumatoid arthritis. Then second thing is a dysregulated hypercitrullination. So, we knows that what is citrullination, and once we knows that there is a something what is called, what we call as a citrullination, then there are two things. One is a normal citrullination process. Second one, pathological. So, normal citrullination process means what? Physiological citrullination. If there is any pathological change in the mechanism of the citrullination, then there can be hypercitrulination or the hypocitrulination. So, accelerated citrulination or decreased citrulination. So, from the viewpoint of this rheumatoid arthritis, is caused by the hypercitrulination. There is a accelerated citrulination which is responsible, which has been considered the responsible factor for the rheumatoid arthritis. And lastly, third point is a mutated HLA-DRB1 gene. Here, when I say mutated HLA-DRB1 gene, here I expect a polymorphism. Okay, so mutated means what? Polymorphism of a HLA-DRB1 gene. It has been considered as a responsible factor for the rheumatoid arthritis. Now, all these factors they have been considered as a etiological factors for this rheumatoid arthritis. And here one thing we should know, or very important, that none of the factor alone can cause this disease. So, anybody suppose having environmental factors are present, that he is having a smoking and mucosal infections are there, will it cause rheumatoid arthritis? The answer is no. Suppose anybody has got hypercitrulination, is this factor alone can cause rheumatoid arthritis? The answer is no. Or this polymorphic HLA-DRB1 gene is present, will it be responsible for the rheumatoid arthritis? The answer is no. So, none of the factor alone can cause this disease. It is a combination of these etiological factors which can cause this rheumatoid arthritis. So, patient could be smoking, he is having a habit of smoking. This patient also has got a hypercitrulination, and also there is a presence of a HLA-DRB1 gene, polymorphic HLA-DRB1 gene, or a mutated HLA-DRB1 gene. The combination of this factor will cause this rheumatoid arthritis. Now we will see what exactly or how exactly this smoking is responsible for this disease. We will also see how hypercitrulination can cause this rheumatoid arthritis, and also we will see what is the polymorphism that is concerned with the HLA-DRB1 gene, and also we will see some additional information regarding all these etiological factors. Because so as to understand this disease, it is very important that we have a complete knowledge of these etiological factors, so that we can understand what is the how this etiopathogenesis that leads to this disease. So, one by one we will see the details of all these factors, and then we will proceed with the other things. Now, let us see some details regarding these etiological factors. So, here in this flow chart, smoking and the infection Okay, so when we say smoking and infection, they are causing the inflammation, means inflammation of the respiratory mucosa or the mucosal tissues. So this smoking and these infections, maybe smoking alone or in combination with the infection, it is causing inflammation of the respiratory mucosa, and because of the inflammation of the respiratory mucosa, there will be increased pad activity. So what is this pad? Pad, it is an enzyme, peptidyl arginine deaminase. Okay, peptidyl arginine deaminase. The activity of this enzyme is increased because of the inflammation of the respiratory mucosa, and this increased pad activity, it leads to increased citrullination or the hypercitrullination. Now same thing, let us see here how this smoking, it causes increased pad activity. Let us see here. This smoking, it is causing chronic inflammation of the lung cells. Okay, so the cells which are present in the mucosa, they are getting chronically inflamed because of the smoking, and because of this chronic inflammation, it is leading to increase in the PAD2 and PAD4 activity. So this peptidyl arginine deaminase 2 and peptidyl arginine deaminase 4, these two variants of these enzymes, they are getting activated, and because of that it is leading to the process of the hypercitrullination. Also, this smoking is causing the cell damage and the apoptosis of the lung cells. So, this smoking is causing the cell damage and the apoptosis of these lung cells, and because of that it leads to raised cellular calcium levels, and because of this raised cellular calcium levels it is causing increased pad activity. Okay, so the pad activity, it requires a calcium, and here because of the raised cellular calcium, this pad activity is increased, and this increased pad activity is again it is causing hypercitrullination. So it is a accelerated pad activity because of the chronic inflammation, and accelerated pad activity because of the cell damage and the apoptotic process. They are leading to the hypercitrullination process in the lung. Now we will try to see what exactly is the citrullination, what exactly happens when we say that the protein is citrullinated, and also we will see some other details regarding the citrullination, so that our concept will be cleared. Now we are going to see the mechanism of the citrullination, what exactly happens with the citrullination process. This understanding this citrullination is very important because it is one of the etiological factor which is concerned with rheumatoid arthritis, and so as to understand the exact pathogenesis process, we should have a clear concept in your mind regarding the citrullination. So before we could see this citrullination, first we should have some basic knowledge. So let us see this box here. So 20 standard amino acids are present in our body. We know that there are 20 standard amino acids, and these amino acids they combine with each other in a particular sequence so as to produce different kind of functional proteins. So we are getting different kind of functional proteins. How we are getting? These 20 standard amino acids, they are combining in a particular sequence, and we are getting different kind of functional proteins because of that. Arginine is one of the amino acid. So of these 20 standard amino acid, arginine is one of them. Why arginine we are taking here? See here. In the process of citrullination, this arginine is converted into citrulline. In the process of citrullination, arginine, which is one of the amino acid, it is converted into citrulline. More specifically, peptidyl arginine is converted into peptidyl citrulline. And this process, the conversion process of arginine to citrulline, this PAD enzyme is very important. Okay. So, PAD enzyme, this enzyme is converting this arginine into citrulline. We have seen this PAD enzyme a few minutes back. And the byproduct during this process is water and the ammonia. So, water and the ammonia, they are the byproduct when the arginine is converted into citrulline, and this mechanism is stimulated by this enzyme what we call as a peptidyl arginine deaminase, that is a PAD enzyme. So, once we know this, let us now see citrullination. In the process of citrullination, nitrogen atom in the arginine side chain is replaced by the oxygen atom. In the process of the citrullination, what exactly is happening? See here, nitrogen atom in the arginine. So, arginine we have just seen that is the amino acid, and the nitrogen atom in this amino acid is replaced by what? Oxygen atom. So, see here, here I have drawn peptidyl arginine here, and this is a peptidyl citrulline here. Now, we want this arginine to be get converted into citrulline. And now, for when I say that arginine is getting converted into citrulline, the nitrogen in this arginine is getting replaced by the oxygen, so that now we can call it as a peptidyl citrulline. So, see here, this nitrogen NH2 which is present in the side chain is getting replaced by oxygen. Okay. So, this nitrogen here is getting replaced by the oxygen, and as a byproduct we are getting water and the ammonia. So, very easy to understand the mechanism if we go like this. So, I just repeat again. So, we have a 20 standard amino acid, arginine is one of them. These amino acids combine in a particular sequence to give us a different kind of a functional proteins, and in the process of citrullination, arginine is getting converted into a citrulline under the enzyme peptidyl arginine deaminase, that is a PAD enzyme in short what we say, and the byproduct is a water and ammonia. In the process of citrullination, nitrogen atom, chemically see this nitrogen atom in the arginine side chain, we have seen this nitrogen atom in the arginine side chain is replaced by the oxygen atom. So, here we have a citrulline, nitrogen is replaced by the oxygen atom here, and this is what the citrullination is. Now, this citrullination we can divide grossly into two types, two categories, physiological citrullination and the pathological citrullination. So, it means that the citrullination is a normal process in the body. Okay. So, once we say that there is a physiological citrullination, meaning that citrullination is normally happening in our body. And what are the examples of the physiological citrullination? They are keratinization, myelination and the gene regulation. There are certain other things also, but for example purpose just I have mentioned the three here. So, when we are talking of the physiological citrullination, what are the examples? Keratinization, myelination and the gene regulation. We will see the details in a few minutes, but for the moment just we have to know that the citrullination we can grossly categorize into two ways, physiological and the pathological. In the physiological we have these examples here. What is the meaning of this pathological citrullination? So when we says that there is a pathological citrullination, we can grossly divide into two: hypercitrullination and the hypocitrullination. Hypercitrullination simply means the citrullination process is accelerated, and the hypocitrullination means citrullination process is decreased. Okay. So the simple meaning of the hypercitrullination, that is the acceleration of the citrullination, and the hypocitrullination, that is citrullination process is decreased. So physiological citrullination is a normocitrullination. The citrullination process is going on with the speed that is normally it should takes place. If the same process, if it is accelerated, it is hypercitrullination, and if the same process is decelerated or decreased, then we can say hypocitrullination. We will see the details of this. Now the disease which we are talking right now, rheumatoid arthritis, it is an example of a hypercitrullination. In the rheumatoid arthritis, we are getting hypercitrullination, and this hypercitrullination is one of the etiological factor. So in the beginning, what I have said is citrullination is very important. Understanding citrullination is very important because it is one of the etiological factor. And when we are talking of a citrullination, as far as rheumatoid arthritis is concerned, this rheumatoid arthritis we are getting, or we are getting a disease rheumatoid arthritis basically because of the hypercitrullination, isn't that? So hypercitrullination, hypocitrullination, this physiological citrullination, we have keratinization, myelination, gene regulation. We will see this in a detail in a few minutes. So I hope that you got at least the concept of what exactly happens in the process of the citrullination, and chemically we says that this nitrogen atom in the arginine is replaced by the oxygen atom. Now we see information regarding these citrullinated proteins, and we will see the names of the proteins and the sites of these proteins which undergoes the process of citrullination. So one by one we will go through these proteins. So first one, structural and cytoskeletal proteins. Under this category we have this filaggrin and keratin. So if you see the chart, here we have a name of the protein filaggrin and keratin, and these two proteins they are the part of the process what we call as a keratinization of the skin. So this filaggrin and the keratin, they undergoes physiological citrullination, and in this physiological citrullination this keratinization of the skin is there. So these two proteins, they involved in the process of keratinization of the skin, which is a physiological citrullination. Then one more protein is a vimentin there. This vimentin, this is here, I just put a dash here with these categories as far as this physiological citrullination is concerned, because basically this vimentin, when it undergoes citrullination, most of the time it is a pathological one, and in the rheumatoid arthritis this vimentin undergoes a pathological citrullination. We knows that this vimentin is present in the filaments there, which is supporting the skin structure, and this protein undergoes citrullination in the pathological process. And in this process, in this pathological condition, the disease is a rheumatoid arthritis. Then next one is a blood clotting and the matrix proteins. So under this category we have a fibrinogen there, and this fibrinogen, which is a blood clotting factor. When we are talking of a physiological citrullination with the fibrinogen, it is involved in the wound healing, So the clot which is formed during the wound healing, it is a part of this physiological citrullination with a fibrinogen. And when we are talking of a pathological citrullation with the fibrinogen, we get it in a disease what we call as a DVT, that is a deep vein thrombosis. So in a deep vein thrombosis, this fibrinogen undergoes a pathological citrullination. Now regarding this collagen type 2, it is a part of a physiological citrullination as well as pathological citrullination. I have not mentioned it here, but this collagen type 2 is also taking part in a physiological citrullination as well as a pathological citrullination. In the physiological citrullination, what we call as a organization of the cartilage, this it is concerned with the organization of the cartilage structure there. When we are talking of a physiological citrullination of the collagen type 2. And regarding the pathological citrullination of the collagen type 2, it is taking part in the pathogenesis of rheumatoid arthritis. So, if there is a pathological citrullination of the collagen type 2, we have the example of rheumatoid arthritis. Then next one is a nuclear protein, that is a histone. This histone which is binding this DNA there, this histone it is taking part in the physiological citrullination, or it is also part of a pathological citrullination. When we are talking of a physiological citrullination, this histone is there for the gene regulation. So the expression of the gene, it depends upon this physiological citrullination process. So for the expression of the gene, it requires this protein to undergo physiological citrullination. And regarding this pathological citrullination of the histone, various kind of a CA what we are getting, it is because of this pathological citrullination of the histone. So the histone is a part of the various malignancies we are getting, and this pathological because this histone is undergoing this pathological citrullination in these various conditions there. Then the next protein is a neurological protein, what we call as a myelin basic protein. In short, we call it as a MBP, myelin basic protein. And this protein is again involved in both these categories, physiological citrullination as well as pathological citrullination. When we are talking about physiological citrullination with this MBP, myelin basic protein, it is there with a CNS development. So this CNS development, this MBP is a very important protein, and this citrullination is helping with the CNS development. Then with MBP, this pathological citrullination is concerned, the demyelination what we are getting, the demyelination means myelin sheath is getting under the process demyelination, and this demyelination of this myelin sheath we are getting a disease what we call as a MS, means multiple sclerosis. So multiple sclerosis is the example of the demyelination, and this demyelination is what is happening with the demyelination. This MVP is getting under a pathological citrullination process to cause demyelination. Then next protein is a enzyme alpha enolase. This alpha enolase, this is we know that it is an important enzyme for the glycolysis process, and this alpha enolase is undergoing, when it is undergoing a physiological citrullation, it is causing myelination. It is responsible for the mechanism of the myelination. And when we are talking of a pathological example of this alpha enolase or pathological citrullination of the alpha enolase, we are getting in a rheumatoid arthritis. So if you see in all, there are few proteins which are only involved in the physiological citrullination. There are few proteins which is involved with the pathological citrullination. and many of these proteins they are the part of the physiological as well as a pathological citrullination. So when we says that these proteins which are involved in physiological as well as a pathological citrullination, when they are taking part in the physiological citrullination, the process of the citrullination is under the control, or it is taking place in a controlled manner. But when this process is accelerated, or when this process is decelerated, or we can say when this process, mechanism of citrullination is getting accelerated or mechanism of citrullination is getting decelerated, then it becomes a pathological citrullination. Okay, so those proteins which are involved in a physiological and both pathological citrullination, when this citrullination is occurring in a controlled manner, it is a physiological citrullination, and when this citrullination is accelerated or when it is decelerated, it becomes a pathological one. So this fibrinogen, this histone, this MBP, this alpha enolase, these proteins they are the part of a physiological as well as a pathological citrullination. So I think you got the point what I would like to say. We will go for the next information. Now here we will see the difference between physiologically citrullinated proteins versus pathologically citrullinated proteins. So physiologically derived citrullinated proteins versus proteins derived of dysregulated citrullination, or we can say pathological citrullination. So what is the difference? See here, as far as the structure of these proteins is concerned, so structure of the physiologically derived citrullinated protein and structure of the pathologically derived citrullinated protein, this structure is the same. So there is no difference in the structure of the proteins, whether it is physiologically derived or it is pathologically derived. And as we know, in the process of citrullination, arginine is getting converted into a citrulline under the influence of the PAD enzyme, and in the physiological citrullination mechanism and the pathological citrullination, both these conditions we are getting conversion of the arginine into the citrulline. But when we are talking of a chemical impact, there is a difference in between these physiological citrullinated proteins versus pathological citrullinated proteins. So what is the difference, or what is the difference in the chemical properties of a physiological citrullinated proteins and the pathological citrullinated protein? See here, these pathologically citrullinated proteins, there is a widespread loss of a positive charges. Okay, so these proteins which are being produced with a pathological citrullination mechanism, there is a widespread loss of the positive charges as compared to physiologically citrullinated proteins. So here in the pathologically citrullinated protein, there is a widespread loss of the positive charges. Because of that, there is a isoelectric point shift towards the acidity. Okay, their isoelectric point is shifting towards the acidity, and there is a increase in the hydrophobicity of these molecules. Okay, these pathological citrullinated proteins, they are more hydrophobic, and also there is an increased protein instability. So they are unstable as compared to physiological citrullinated proteins. These pathological citrullinated proteins, they are more unstable and altered self epitopes. Okay, so in these pathological citrullinated proteins, they have altered self epitopes. So the point is, this whether it is a physiologically citrullinated protein or a pathological citrullinated protein, structurally they are the same. Structurally there is a no difference. But when we are talking of the chemical impact, there is a difference. There is a great deal of difference in between physiologically citrullinated proteins and the pathologically citrullinated proteins. We are discussing various etiological factors which are concerned with rheumatoid arthritis. So, in the beginning only we have seen three factors are responsible for the rheumatoid arthritis, and we have already discussed two factors. First factor is smoking. How the smoking is concerned with the rheumatoid arthritis, that we have already seen. Also we have seen that how these citrullinated proteins they are the part of the rheumatoid arthritis, especially dysregulated citrullination, how it is responsible for the rheumatoid arthritis. And lastly we will see this factor what we call as a HLA-DRB1 gene, how this gene is a part of the rheumatoid arthritis. So see here, this HLA-DRB1 gene, we know that it produces a beta chain. It produces a beta chain of the MSC class 2 molecule. So this HLA-DRB1 gene, it produces a beta chain which combines with the alpha chain, and this combination gives us complete MSC class 2 molecule. So this major histocompatibility factor 2, it is formed of two chains, alpha chain and the beta chain. Of these two chains, the beta chain is formed by HLA-DRB1 gene, and this gene is present over the chromosome number 6. Now a groove is present over MSC2 molecule for the antigen. So this MSC class 2 molecule, it has got a groove over it to combine with the antigen. HLA-DRB1 gene, because of the polymorphism property, sometimes may produce variations in the beta chain. I again repeat, HLA-DRB1 gene, because of the polymorphism property, because of the polymorphism, sometimes it may produce a variation in the beta chain. Now first we will see what is polymorphism, and then again we will come here. So see here, polymorphism. So polymorphism, it is a common occurrence of two or more gene variations in a population. So if you take a group of a population there, then in that particular population, different individual might be containing a different kind of a gene for the same trait there. Okay. So this HLA-DRB1 gene, it is a polymorphic. That means that it has got two or more genetic variations in the population. And when we say genetic variation, a variant must be present at least 1% in the population. So when we are talking of a different variation, if you take any variation, it should have existence of at least more than 1% in the population. So suppose when we say that HLA-DRB1 gene is a polymorphic gene, that means that this HLA-DRB1 gene, it has got some genetic variations there, and suppose for example, or we can say for the sake of the convenience, rather than examples, for the sake of the convenience, suppose this HLA-DRB1 gene, it has got three variations. okay, and this variation A, variation B and the variation C. It may be possible that this HLA-DR1 gene variation A may be present in 90% of the population, and then HLA-DR1 gene B type of a variation may be present in 8% of the population, and HLA-DR1 gene C type of a variation may be present in a remaining 2% of the population. Okay. This is just for the sake of a convenience. It is actually the things are not like that, but for the sake of the understanding, suppose this polymorphic HLA DR1 gene, okay, it has got type A, type B and types C. These three variations are there. Suppose this variation is there for 90% of the peoples, and this variation may be present in 8% of the patient, and this variation may be present in 2% rather than patient. We can say population, 90% of the population, it might have been type A, 8% type B and 2% type C. This is just for the sake of the convenience we are looking at this. So these different variations of the HLA-DR1 gene, it is present in this population, suppose, and it may be possible that this type C, which has got 2% variation, it may be producing a disease there, and this remaining 98% of the population, they may be free of any disease. So what does it mean? See here, this HLA-DR1 gene, because of the polymorphism property, sometimes may produce a variation in the beta chain or a groove which is susceptible for the autoimmune disease. So this HLA-DR1 gene, which is a polymorphic gene, it is producing a different kind of a beta chain. Because of the polymorphism, it is producing a different types of a beta chain. But some beta chains which are formed because of a particular type of a genetic variation, they are producing such a type of a groove over this MSC class II molecule where it is susceptible for the production of the antibodies, or it is susceptible for the autoimmune disease, isn't that? So here in this case, this type A variety, it is producing a beta chain which is not responsible for the autoimmune disease. Type B variety, it is producing again a beta chain, but again this beta chain is not producing any types of disease. But type C, it is producing a beta chain. This beta chain is susceptible for the autoimmune disease there, because this beta chain, when it is forming a groove, this groove is susceptible for the autoimmune disease. Now read this again. HLA-DR1 gene, because of the polymorphism property, sometimes may produce a variation in the beta chain or the groove which is susceptible for the autoimmune disease. So this groove which is formed over this any MSC class II molecule, now here this groove, when we says that this particular groove is susceptible for the autoimmune disease, then this groove we called as a shared epitope. Now this groove, of course, it is a binding site for what? Binding site for the antigen. Groove here we means that it is a binding site for the antigen. Normally this binding site, when it is formed, it is not susceptible for the this autoimmune disease, but this but in some group of peoples, or in some percent of the population, where because of the polymorphic property, some of the genetic variations of this HLA-DR1 gene, they may produce such a kind of a beta chain, which when forming a groove, this groove is susceptible for the autoimmune disease, and this groove which is susceptible for the autoimmune disease we call as a shared epitope. We know that epitopes are present over the antigen. Here this shared epitope, when we say shared epitope, this shared epitope is present over MSC class 2 molecule. And why we are calling it as a shared epitope? Because it is a groove which is susceptible for the autoimmune disease. So this is how this shared epitope, this word is very important with rheumatoid arthritis. In rheumatoid arthritis patient, shared epitope is present over this MSC class 2 molecule, and this shared epitope by definition we can say that it is a antigen binding groove which is present over the MSC class 2 molecule, and this particular groove is susceptible for the autoimmune disease, and that is why we call it as a shared epitope. So this is how we can explain this how this HLA-DRB1 gene is responsible for the it has been responsible for the rheumatoid arthritis. So we have already seen the description regarding the etiological factors which are responsible for the rheumatoid arthritis. These shared epitopes which I have just seen, susceptible sites for the autoimmune disease present over the HLA-DRB chain. Dysregulated citrullinated proteins and the smoking. So these three factors, shared epitopes, dysregulated citrullinated proteins and the smoking, these three factors they activates the immune system, and in the end this immune system they starts the production of the antibodies. We have learned that in the process of the citrullination, arginine is getting converted into citrulline. This arginine, it is a positively charged, whereas this citrulline protein, it is a neutral. This arginine protein, it is a positively charged, whereas the citrulline is a neutral. And third thing here, antigen presenting groove. Antigen presenting groove in the sense this we can consider here as a shared epitope. So this antigen presenting groove or shared epitope, it is been a positively charged. Okay, it is a positively charged, and this positively charged shared epitope, this arginine, it is again been a positively charged and a citrulline is a neutral. Now why it is important to know that what are the charges over this? See here, normally when this arginine is getting converted into citrulline, so the process what we call as a physiological citrullination. In the process of the physiological citrullination, this arginine, it is getting converted into citrulline. In this mechanism, there is a loss of positive charges. We know that when arginine is getting converted to citrulline, this process is occurring under the influence of the PAD enzyme. PAD enzyme is acting as a catalyst, and with this catalyzer property of this PAD enzyme, this arginine is getting converted into a citrulline, and in this mechanism there is a loss of positive charges. So arginine, it is a positively charged, we are getting a citrulline which has been a neutral one. Okay, it is a neutral one. Now we are talking of the event what we call as a dysregulated citrullination. Okay, dysregulated citrullination or uncontrolled citrullination. See what is happening here. Also this arginine is getting converted into citrulline. Okay, so in a dysregulated citrullination also we are getting citrulline from the arginine with the same this pad enzyme activity. But what is the difference? So there is structurally no difference. Here also we have a citrulline, here also we have citrulline. There is structurally no difference. But see what is happening with the chemical properties here. When there is a dysregulated citrullination, there is a more loss of positive charges. So as compared to physiological citrullination, in a pathological citrullination, we have a more loss of positive charges, and therefore the citrulline molecule which we are getting because of this dysregulated citrullination, this citrulline molecule is a more negative as compared to physiologically formed citrulline molecule. I repeat again, this pathologically formed citrulline molecule, it is a more negative as compared to physiologically formed citrulline molecule. Now what is the importance of this? See here, we know that this antigen presenting group, or we can say shared epitope also, it is positively charged. It is positively charged there, and this antigen presenting group, it can combine with this physiologically formed citrulline because it is a neutral. So there will be a good amount of bonding in between positively charged shared epitope or antigen presenting group and the citrulline, okay, which is a physiologically formed citrulline, of course. But when this antigen presenting group, it is combining with a pathologically formed citrulline, this bonding is more strong. Why? Because this pathologically formed citrulline is more negative. So this positive group combines more strongly with this more negative group because of the opposite forces. This positive group can combine with more negative citrulline with a firm bonding as compared to the citrulline which is physiologically formed, isn't that? So when this antigen presenting group which is a positively charged, when it is combined with a physiologically formed citrulline, this bonding is a good one, no doubt. But when this antigen presenting group, it is combining with a pathologically formed citrulline, this bonding is very strong as compared to this bonding. Why? Because this citrulline here, it is a more negative as compared to this, and because it is more negative and here it is a positive, this bonding is very firm there. Now what is the importance of all these things? See here, results in, so this pathologically formed citrulline, when it combines with the antigen presenting group, see what happens, results in a stronger bond formation between antigen presenting group and a proteins produced of a dysregulated citrullination. We have seen why the strong bond is formed, because it is more negative here. Deceives the immune system. So this firm bonding, it deceives the immune system, because normally the bonding should be good enough, but here the bonding is very strong, and therefore our immune system is getting deceived and it start producing the antibodies against this combination, isn't that? So why the antibodies are produced in whole this mechanism? It is because of this difference in the chemical properties. Structurally they are the same. Here it is arginine citrulline, here it is also arginine citrulline. Structurally there is no difference, but because of the difference in the chemical properties, this immune system is getting deceived and it is starting producing antibodies against it.